इतना लंबा पॉइंट चलेगा बन जाएगी। हार गया जीत गया तो हार गया क्या करते हैं काटेंगे तो एकदम ठीक करेंगे होना भी चाहिए लाइफ
Okay, let's get started for the day. Welcome everyone to the week 8 molecular biology lecture on the PMRF NPTEL session. So this week we are going to deal majorly with different molecular biology techniques such as northern and southern blotting as well as western blotting. As you would know that the PMRF-NPTEL lectures are designed in the way so that we can go through the previous year's run of the course and the quizzes so that we have a better understanding on how to answer the quizzes this year and also get a better understanding of the concepts behind the entire course. I will briefly introduce myself. My name is Anuvab Chatterjee, and I'm a PMRF fellow and PhD scholar at IIT Kanpur, and I will be taking this course. So, how we do this is that I will show you a question from the previous year's run of the course, and I will give you some time to try and solve it, and then we will solve it together when I show you the concepts behind the question. Okay, so let us get started with the first question of the day. The question is to precipitate the genomic DNA from aqueous phase, blank is commonly used. The options are water, acetic acid, absolute alcohol, and Tris buffer. I will give you a moment to think about it. You can put what you think is the correct option in the chat. Okay, so let us look at it. So there are different blotting techniques we use in molecular biology. The major ones that were previously used some time back and are fundamentals of the techniques used in our modern molecular biology labs include the southern blotting, which was the first one that was discovered, which is used to probe for DNA. So if you want to probe for a specific DNA fragment or a sequence or a gene, and if you want to know from a pool of DNA if your particular gene is present or not, then you use something known as southern blotting. A similar technique is known as northern blotting, where you want to find if a specific piece of RNA is present or not. This is majorly used to detect if your mRNA of interest is being expressed in the cells that you are probing. And if, say, gene A is present, then you should see a blot. Third is when you try to figure out if your tissue or cell of interest have a particular protein, which is known as the Western blot. And finally, there is carbohydrate and Eastern blotting. So why do we call all of them blotting? Because all of them are done on something known as the nitrocellulose membranes. So they look very similar to a piece of paper, which is specifically made to bind to either DNA, RNA, or protein. And then you use some kind of colorimetric reaction to essentially highlight the part where your DNA, RNA, or protein of interest is there. So it looks like essentially a blot or a darker patch of just it essentially looks like a darker patch of a blot, like when you would have from a gel pen or something on a paper. Essentially, that is why it is known as blotting techniques. The first that we are going to talk about is southern blotting, which I said is used for DNA. So for southern blotting, you start from genomic DNA. So you take your cells, you separate out the DNA, that is the first step. then you cannot probe the entire DNA at the same time because you would need be- because you would need smaller fragments. You do generate smaller fragments using restriction dilatation enzymes. So there are essentially the entire genomic DNA is now fragmented into small pieces. Then, instead of having them all together in a test tube, you then load them into something known as an agarose gel. I imagine that most of you would have an idea of what an agarose gel is. Agarose gel is something like this. It is a gel on which you can run DNA and perform something known as gel electrophoresis, which separates the DNA fragments depending on their weight or how big they are. So essentially, for genomic DNA, we would imagine a lot of bands coming all the way from the top to the bottom. And then you transfer the entire DNA fragments onto our nitrocellulose membrane, which is looks like a piece of paper, just as I said. And on top of the nitrocellulose membrane, you hybridize it using a radio-labeled probe. This probe is specific to your gene of interest and it is complementary to the gene of interest. So the moment you let it hybridize, it will go and base pair and it will attach only to the region where your gene is present on the nitrocellulose membrane. And since the probe is radio labeled, you can do something known as autoradiograph, which is essentially you having a silver nitrate plate on top on opposite to it, and then you shoot it with X-rays. And wherever there is any radio labeling, that will essentially darken. And essentially, you will be able to figure out where on the nitrocellulose membrane your piece of DNA was. So this is a big complicated process. We will take it bit by bit and understand more of it. So this is a video which describes in basic terms what exactly a southern blot is, and we'll take it from there. I will show you this video which is available on YouTube and the reference is given onto the slides as well. Just let me know if you can hear the sound.
So in this video, I'm going to be
The sound is audible.
talking about something known as a southern blot. So a southern blot basically allows you to visualize a specific piece of DNA that you're interested in. So let's imagine that we have a cup and it's filled with DNA. So it's got just a whole bunch of DNA inside. And there's just lots and lots of this DNA. And let's imagine that I'm specifically interested in one gene. So let's imagine that I'm interested in gene A. And I want to see if gene A is inside of this cup, if it's inside of this long piece of DNA. Now, in order to figure out whether or not gene A is inside this cup, basically we have to do this process known as a southern blot. And we'll break it up into a couple of different steps. So step one, what we're going to do is we're going to take this DNA and we're going to cleave it. So take the DNA and cleave it. So let me draw that out. So we're going to take this big old strand, we're going to remove it outside of the cup over here. So we've got this big strand, and we're going to cut it up. We're going to expose it to enzymes that will basically cleave the DNA in a whole bunch of different parts. And that'll result in lots of these smaller pieces of DNA. And so that's basically the first step. So we've got a bunch of small little pieces of DNA. Now, step two, what do we do? Well, what we're going to do is we're going to take all of these tiny little DNA fragments and we're going to run them on the gel. So specifically, we're going to do a gel electrophoresis on these DNA fragments. And I made a video on gel electrophoresis, so if you want to refresh, you can watch that video. But basically, the gel electrophoresis will help us separate these DNA fragments based on size and based on charge. So let's just diagram that out. So we're going to take these DNA fragments and we're going to run them on a gel. So let's imagine that this is the gel, and we add the DNA fragments to different wells. So the fragments are going to move down the gel, and they're going to basically be separated based on size and based on charge. So we're going to have these fragments separated, like so. So now we've got this gel and we've got the DNA fragments separated by size on this gel. So the next step, step number 3, is basically we're going to take this gel and we're going to transfer it to a filter. So transfer the gel onto a filter. And what the filter will basically allow us to do is it'll allow us to visualize, because this gel is very flimsy, so we want to transfer it onto a filter. So what we'll do is we'll take a filter that's basically the same size as the gel, and we're going to basically just put it right on top of the gel for a little bit, and the fragments will basically transfer onto the filter. So now we're going to have a filter with these fragments, and the filter is a lot sturdier than the gel. So this is the filter, and I'll just write that down over here, and this over here is the gel. Okay, so the next step, step number 4, is we're going to take the filter and we're going to expose it to a radio-labeled piece of DNA. So expose to radio-labeled DNA. Now this radio-labeled DNA is going to be the complement to our gene of interest. So we're interested in finding out if gene A is present in this massive DNA over here. So what we do is we're going to take the complementary sequence to gene A and radio-label it and expose it to this filter. So let's imagine that the radio-labeled piece of DNA is this pink piece of DNA. And let's imagine that we do have gene A. So let's imagine that this piece of this DNA fragment was actually gene A, our gene of interest. So what's going to happen is when we expose the radio-labeled DNA to this filter paper, it's going to anneal to our gene of interest. So we're going to have this radio-labeled piece of DNA stuck to this DNA fragment, which is its complement. So in order to visualize it, in order to visualize this radio-labeled piece of DNA, we have to do the 5th and final step, which is expose the filter to an X-ray film in order to visualize the radio-labeled probe. So expose to X-ray. And the X-ray, basically, it'll shoot a bunch of X-rays, and since this piece of DNA is radio-labeled, it will pop up on the X-ray film. So we're going to have a film, and we'll draw that film over here. So we'll have this film, and basically the only thing that'll pop up is this fragment over here, and that fragment will have a control, and we'll be able to say, okay, well, since we have this fragment, it's basically the radio-labeled piece of DNA, and since we see the radio-labeled DNA, it means that it was bound to this gene A, which means that gene A was in this cup of DNA.
Okay, I hope that clears some of what we are doing here. Uh, I hope that was. Does anyone have any questions about the basics of Southern blotting? If you have, we can go through it again. Otherwise, we will move forward to how exactly to go about the first step, which is.
So in this video, I'm going to be talking about something known as a
That's right. And if I want to go to the next page, yes. Okay. So as we saw, Southern blotting can be divided into multiple steps. First step is isolation of the DNA that you need to blot. Then you do something known as a restriction digestion, which is using restriction digestion enzymes to cleave the genomic DNA, which is huge, into small fragments. And then you do the entire business with nitrocellulose membranes and then probing it with a radio label probe. So here we are going to discuss the first step, which is genomic DNA isolation. Okay. So for genomic DNA isolation, first you have to have cells or tissues that you want to take the DNA out of. You take them and you do something known as lysis of the cells. So you lyse the cells so that all of the cells essentially give up the entire genomic DNA and then are ruptured. So that, and this rupture is done enzymatically or using a detergent such as SDS, which completely opens up the cell membrane so that the genomic DNA is now open to the buffers that you add. This is followed by incubation with a particular enzyme such as proteinase K, as well as more SDS. so that you denature all the proteins. The idea here is that the cell will contain a lot of biomolecules such as DNA, RNA, lipids, and proteins, as well as carbohydrates. And you want to systematically remove all of these so that you only have very pure genomic DNA at the end. So the first thing you remove is the protein. You use something called proteinase K, which will digest and essentially cleave all the proteins, and SDS, which will denature all the proteins. So proteins which are bound together in huge complexes will all get denatured into single strands, which will then be cleaved by proteinase K. This also helps in removal of proteins from genomic DNA. So we have learned that histones and the histone complexes actually bind the DNA into the chromosomes. So if you want to extract the genomic DNA, most of the time you will get a lot of histones coming up as well because they are very tightly bound. But when you use a combination of SDS to actually denature the histones, so they let go of the DNA and then proteinase K to cleave them into small fragments and essentially destroy them, then you only will have the genomic DNA from the entire solution. So you digest and remove all the proteins in this step. Next, what you do is that you precipitate using absolute alcohol. So this is a very interesting step. So essentially the solution at this moment has different components such as your proteins, your lipids, carbohydrates, everything else. You use the solubility of DNA, which is going to be polar in nature, in order to take it out of solution. So what do you do? So ethanol, so normally DNA we have seen is a double helix structure, but in solution it is soluble because it is stabilized by water molecules. All these dots that you see right here is known as a hydration shell. Essentially, the DNA has negative charges all along its backbone, but these negative charges, these close together, are unstable. But when you dissolve DNA in water, what happens is that the hydrogen molecules in water actually cover the entire DNA, essentially covering the negative charges and neutralizing it. This stabilizes the DNA and actually allows the DNA to be soluble in water. This is how solubilization works, and depending on how well a particular solvent can create this hydration shell around an ion or a molecule shows how well it can dissolve something. So where does ethanol come into this? So ethanol, what it will do is that it will decrease the solubility of DNA. Since ethanol also has a OH group and can form hydrogen bonding with water, but it is not as soluble as water. So when you add 70% ethanol to the solution containing different proteins, DNA, lipids, carbohydrates, it will actually cause the DNA to be unstable because the DNA now has many exposed negative charges, and DNA will try to come out of solution as precipitates. So once it comes out of the solution as precipitate, you can centrifuge the tube and you will have a very tiny pellet of DNA, and that should be a very pure DNA which does not have any other impurities. So ethanol is thus used for precipitation of DNA. The next step you do is that so this absolute alcohol precipitation and chloroform phenol purification steps can be reversed. So you treat the entire solution with chloroform and phenol. Chloroform and phenol, you know, are organic solvents. Chloroform is mostly organic. Phenol is polar with the OH group. What these two is chloroform will bind to all the lipids that are present in the solution, and chloroform will dissolve them. So all the lipids will try to go into chloroform. And phenol, what it will do is that it will denature whatever proteins are left and cause the proteins to get dissolved in phenol. When you centrifuge the tube now, the solution, which originally had DNA, water, all the cell impurities, now will form two separate layers. One, which will be a layer of just water, where there only will be DNA or other nucleic acid, which are polar enough to be dissolved in the aqueous phase. And the bottom layer will have a mixture of chloroform and phenol, where in the chloroform, there are various lipids dissolved, and the phenol is where the protein is dissolved. This step is also accompanied by addition of isoamyl alcohol to this mixture of chloroform and phenol. Isoamyl alcohol is a ambivalent group, so it has an organic part as well as a polar part. So it essentially forms a middle layer between the organic chloroform and phenol layers and the upper aqueous layer. So it looks something like this. So this is your aqueous layer right here. You have your organic layer at the bottom, and in the middle there is this thin layer of isoamyl alcohol. So why do we need isoamyl alcohol? Many proteins are something called ambivalent in nature, where they have an organic as well as a polar group. So they do not dissolve completely in phenol and chloroform, and they try to stay in the aqueous phase. But when you add isoamyl alcohol, it traps all of these ambivalent proteins and traps them in this single cloudy layer in the middle of the aqueous and the organic phase. And then you can just take the DNA, which is dissolved in the aqueous phase, out using a pipette. So you just take all of it out. So major reason of using chloroform phenol is dissolution, dissolving lipids as well as protein and denaturing them, so that you have two different layers, the aqueous and the organic phase. Aqueous will contain the DNA. So essentially you spin again in the centrifuge and take out the aqueous layer. And then you can again do a precipitation with absolute alcohol to remove all the salts that are present. So genomic DNA present in the aqueous phase is again precipitated using absolute alcohol. So you precipitate the DNA again, and only very small amounts of salts should be precipitated with the DNA, because most of anything else that is polar in the cell, such as proteins, has already been taken care of in the previous step. And next, what you do is that you run it on an agarose gel. You will learn what that is in a bit. And in the agarose gel, essentially you run it and it looks something like this. So this is the image of agarose gel. You load the DNA in something known as a well. So essentially a cavity in which you can load the DNA, and you apply an electric field to both ends, negative on this end, positive on this end, and DNA will obviously try to move to the positive end, and depending on the weight of the DNA, it will either move a lot or not at all. And genomic DNA should look like a smear or, or a very solid band at the top of the well, because genomic DNA is huge and does not like to move in electric fields. So it will probably be stuck in the top. So a good preparation of genomic DNA gives an intact band with no visible smear, which we can see right here. So this is how you isolate genomic DNA for the first step of northern blotting. And to precipitate genomic DNA from the aqueous phase, absolute alcohol is commonly used. Any questions? Okay, moving on. In southern blotting, the DNA fragments are separated by using blank, high performance liquid chromatography, capillary electrophoresis, agarose gel electrophoresis, and SDS page. You can put in whatever you think is the correct option in the chat. You can put in A, B, C, D, whichever you think could be the correct answer. Fusion says it is C, agarose gel electrophoresis. Anyone else? Or Rupi also says it is C, agarose gel electrophoresis. So let us look at it. So once you have isolated the DNA, the next step that you do in southern blotting, you fragment the DNA either using sonication, which is you use a probe that vibrates at a very high frequency and that will break up the DNA, or you can do something known as restriction digestion. You take a randomly cutting, you take a restriction enzyme that will produce random cuts throughout the DNA so that it is fragmented into smaller pieces which can run on agarose gel, and you separate. So you use agarose gel electrophoresis. So agarose is a seaweed derived component which when you mix in water, it is generally does not dissolve. But when you heat the water up, then agarose dissolves very well. And when the water now cools down, it sets into a gel. So it will, if you have ever seen gelatin in work or which we use in cooking and also making ice creams, you would know how that works. So essentially, you form a very thick, solid jelly-like structure. And use of agarose is important because agarose forms very uniform, very uniform pore sizes in the gel it makes. So you will have a square gel that will have microscopic holes through which DNA can go through, and that movement is very uniform. So that is why agarose is used, and you make essentially a gel, which is like a jelly-like structure, which is in a square shape, through which DNA can move. As a standard practice, agarose used for southern blotting is ultra pure, and we do something known as staining the DNA with ethidium bromide to visualize it, which we will look at. So this is what a gel looks like. This transparent jelly-like structure right here, and you have these wells at the top, and these wells are where you load the DNA. So this is how you load the DNA with a pipette. And the DNA normally is colorless. You add something known as a loading dye to the DNA, and this is why it appears blue in this image. So blue or purple depending on what kind of loading dye you have. And the reason you use a loading dye are there are various reasons. The first one is that you can see the DNA and you know you are loading it correctly, because otherwise it is completely transparent. You do not know if you are loading them correctly in the wells. The second reason is that DNA, the density of the DNA is very close to water. So if you load the DNA into these cavities, which are known as wells, and you let it sit, DNA will just float out and go all over the jelly. They will not travel through the gel. If they travel through the gel, then we can separate them based on weight. If they just float over in the buffer surrounding the gel, then you do not have any DNA. So you won't be able to visualize anything. You want the DNA to be forced to travel through the gel. And that is why it uses something known as a loading dye. So loading dye binds the DNA and essentially pulls it down to the bottom of the well, so that now when it has to move through the gel. And this you can use high density glucose or sucrose or something. And finally, it also has something known as a tracking dye, and we'll see how a tracking dye works in a bit. But essentially, this is what agarose gel electrophoresis looks like. So this you have your gel, you have loaded them into the wells, and you attach two electrodes, one to the top, which is going to be a negative electrode, and one to the bottom, which is going to be a positive electrode. And then you let electricity flow through the gel as well as the buffer. And obviously, DNA fragments, negatively charged as they will move away from the negative electrode towards the positive electrode. And electrophoresis depends. So how much your DNA fragment will move depends on a parameter known as electrophoretic mobility, and that is proportional to the charge by mass ratio of the DNA. Since DNA is uniformly charged because all the base pairs have one negative charge, it essentially depends now on the mass of the DNA. And it is inversely proportional. Essentially, heavier DNA will be towards the top, and lighter DNA will travel very fast to the bottom of the gel towards the positive electron. So essentially, you can separate now different DNA fragments depending on how heavy they are. So here is another image. You have a negative electrode, you have a positive electrode. The longer and bigger fragments of the DNA will stay closer to the wells towards the negative electrode, and the shorter and smaller fragments will travel very fast towards the positive electrode. And you have different bands essentially. And so essentially your DNA has traveled, but your DNA is again transparent. So how do you visualize it? you use something known as ethidium bromide staining. So ethidium bromide, it is an organic molecule that has two different properties. First, it loves to get in between the different bases in a double helix of DNA. So it does something known as intercalation. So you have a double helix of DNA and hydrogen bonding between the bases. It intercalates in between these bases like this. And secondly, once it intercalates, it interacts with the pi-pi stacking of the different organic bases, the ATGCs. And essentially, due to this interaction, it is now, if you shine UV light on it, it will fluoresce. And essentially, when you shine UV light on top of it, the DNA will now glow like this, and you will have this beautiful bands that are throughout the gel, and you are able to figure out, yes, this band is lighter than this one, and then you compare different things, but majorly the weight of the DNA. Another smart thing that we have people have figured out is that okay, so to figure out exactly how much the size of the DNA is, you need a relative measurement. So how big is this DNA? I don't know because I don't have any measure of how much it has traveled. So what we use are known as ladders. So ladders are pre-digested fragments of DNA which always flow and arrange themselves in a particular order. For example, the ladder shown right here in this image is a 1 kb ladder, and we know what the different sizes are. For example, this fragment is 3 kb, this is 2, this is 500 base pair. So wherever your DNA ends up, you just draw a line along it and you can figure out for this. So if this is 3 kb, this is 2 kb, then this is probably all the bands here are 2.5 kilo base pair big. Similarly, on this, if this is our 5 kb band, then you can say that this particular band is a bit bigger than 5 kb, smaller than 6 kb, around 5.5, and essentially you are able to separate DNA based on the size. So you know how big your DNA is and how much it has traveled through the gel. So this is what you do in gel electrophoresis. And in again southern blotting, the DNA fragments are separated by using agarose gel electrophoresis. Any questions from this? Okay, moving on. In northern blotting, which is what you use to probe for RNA molecules, a blank probe is typically used to detect specific RNA molecules. The options are DNA, amino acids, proteins, or antibodies. Which of the following do you think is used to detect RNA molecules in northern blot? I will give you a second to think about it. phone isn't working. What happened? Okay, we'll reload, join. Hopefully you can hear me better again. Mhm. Okay, I did see multiple A options, which is everyone said that it is DNA probes that are used to probe for RNA molecules. And you are correct, it is DNA probes, Srijani and Oropi, I think, answered. So let us look at it. So, as we have been saying, you use probes to detect the RNA that you have on the gel. So, it is very similar process to just give me a second to close the door. It is a very similar process to how we do southern blotting. You just start with RNA instead, and you make a cDNA out of it, and then you detect the cDNA in the gel and what you use to detect. So for detection of the DNA or RNA molecules, the easiest thing to use is a complementary strand of DNA. So you essentially make the complementary strand of the DNA and elect it base pair to the DNA or RNA of interest on a gel or anything else. And then due to some properties of the particular probe, you can detect it. For our situation, we have a radio label probe. So when you are making your probe, you were putting in particular bases which are radioactive. And as we have been seeing, even with southern blotting, you can use something known as autoradiography to actually develop the signal, and you will see on where on the gel actually your probe is. So making the probe, you can make it in various different ways. One of them could be the random primer method. In this, a random primer is used to anneal to the template. So you have a source DNA and you allow your primers to bind anywhere and everywhere. And then you do in vitro transcription, in vitro replication reaction. Essentially, you allow Klenau fragment, which is a DNA part of a DNA polymerase, to synthesize one part of this probe, and you supply as DNTPs, radio labeled DNTPs. So where whatever small fragments are getting made, all of these have radioactivity in them, and you separate these probes out. And when you probe your northern or southern blot gel, you will be able to hybridize one of these probes wherever your particular fragment is present, and that will be radio labeled. Random primer is so the source double-stranded DNA is denatured to generate single-stranded DNA template. A random primer is added and allowed to anneal to the template strand. It will anneal to random position throughout the sequence in multiple places because it is a random primer. So the sequence is completely random, so it will have chances to bind anywhere. The primer will anneal to the template strand, and the clean now fragment will start to synthesize a DNA strand. Newly synthesized DNA will give short stretches of multiple labeled DNA probes. So this is how you label your DNA probes, and then you can use these probes to detect your DNA or RNA of interest. And this is what the result looks like. So you have essentially this is after you have transferred onto the nitrocellulose membrane, and you have probed using your radio label probes. So wherever your radio label probe will bind, after development by autoradiography, you will see this dark spot on it. So you see only your DNA of interest has been labeled. And probably in untreated control, the amount is very low compared to your treated control, so you can compare it even. And since this particular technique is quantitative in nature, so you need to know the amount of DNA. So if there is a third gel which is even darker than this one, then you can say yes, then there is double the amount of DNA in here compared to here because it is quantitative in nature. So in northern blot, DNA probe is basically typically used to detect specific RNA molecules. Next, blank enzyme is used to get a single cell suspension from liver. The options are trypsin, collagenase, amylase, and lipase. You can put in whatever you think is the correct option in the chat. Srijani says it is B, collagenase. Anyone else? Oropi also says it is B, collagenase. So let us look at it. So when you are, if you would remember in northern blotting or southern blotting, the first step is going to be isolation of your genomic DNA or total RNA, depending on what you are trying to do. But if you are harvesting that from a tissue sample, for example, if you want to investigate the inflammation of pancreas in a mice, you would first have to take the tissue out of the mice, and then you will have to digest the entire tissue. And the entire tissue isolation of DNA or RNA cannot happen from the tissue. The techniques that we use are supposed to be used on cells because tissues are very resistant to all of these different enzymes or different conditions that you use to isolate DNA or RNA. So the first thing you do is known as a collagenase digestion in case of liver or pancreas both. What that will do is that, so how is a tissue this salt? So what happens is that cells themselves are very discrete in nature. They are single entities. But when they come together as a tissue, they release a extracellular matrix and adhere to one another. This extracellular matrix essentially works as a platform on which the cells can sit, and then they adhere to the extracellular matrix as well as each other to form the entire construction of the tissue. Using ECM degrading enzymes such as collagenase, you can break the bonds of the ECM and separate the entire tissue into a single cell suspension. A single cell suspension can be used for different experiments, including isolation of genomic DNA. So depending on the tissue you're using, you can use collagenase, trypsin, or various other, depending on cocktails of proteinase K to separate the tissue into single cell suspensions. Then once the treatment is done, you can separate them into different, so you can separate the cells into different smaller, so you do the more stuff to them so that they form single cells. Then you can culture them, and depending on the culture you can essentially pick out the cells, or even for our interest we can proceed from this step only, where the entire tissue has been disintegrated into single cells to extract genomic DNA. This particular protocol is showing how to handpick islets, which are the islets of Langerhans from pancreas, and study them for insulin secretion. So that can also be done with a similar procedure, but it is much more complicated. We are just interested in a single cell suspension to isolate genomic DNA. If you want to do more stuff, then you can plate them onto Petri plate and then essentially pick out individual islets and then store them. Essentially, you just pick out the islet cells, and then using the islets, you can do multiple different things. You can do insulin tolerance tests, glucose tolerance tests. You can do RNA sequencing upon addition of different conditions and then analyze. But essentially, collagenase enzyme is majorly used to get a single cell suspension from liver. Any questions? Okay. Moving on. Chloroform is actually going to remove the blank part during genomic isolation. The lipid, the DNA, the RNA, or the carbohydrate. Or says it is A, the lipid part. Anyone else? Srijani also says it is A. Okay, so let's look at it. So we have already seen how genomic DNA isolation works. The chloroform part of it is works as an organic solvent, which dissolves the lipids and pulls them down into the organic phase. And that is what it does. And just for a reminder, phenol does the same to proteins. It dissolves them and pulls them down into organic phase when you centrifuge the entire tube. And you should then have two separate phases, the aqueous phase that will contain our DNA and similar polar molecules, and your organic phase will contain your lipids as well as the proteins. So chloroform is actually going to remove the lipid part. Phenol is going to denature the proteins and they will become part of the blank phase, aqueous, gaseous, solid, or organic. You can put in what you think is the correct option in the chat. You can put in what you think is the correct option in the chat. Okay. So as we have been discussing, the chloroform and phenol, they will form the organic phase and the phenol will trap the proteins in the organic phase. Aqueous phase will have the DNA. So the phenol is going to denature the proteins and they will also become part of the organic phase. A smear on the gel is actually an indication that you have blank genomic DNA, which suggests that you have either broken down or degraded the genomic DNA. What does a smear, which is and not a very sharp band on a gel mean? Does it mean that you have amplified, fragmented, isolated, or purified genomic DNA on your gel? You can put in what you think is the correct option in the chat. Rupi says it is B, fragmented. Anyone else? Chanda says it is B. Devadrita also says it is B, fragmented DNA. Srijani also says it is B, fragmented DNA. Okay, so let us look at it. So this is what a gel with DNA's contamination or fragmented DNA look like. You should have very sharp bands such as this, or what you can see in the ladder. Sharp singular bands like this. But if you have multiple bands in the genomic DNA, which you can have, but essentially the smear is this hue of this light hue that you can see throughout the gel. If you had a single sharp band, and you can see the contrast that there is a single sharp band and there is a band followed by a hue. This means that the DNA has fragmented into smaller and smaller pieces and is now all over the gel. This can happen when you're not the this essentially happens when you have a DNA's contamination somewhere down the line. And mostly the most suspected candidate is always the water that you are using for your experiments that is somehow disrupting your entire process and causing the DNA to fragment. Or so replace the water and start over again and you should have better results. But essentially A hue on the gel like this would mean that your DNA is degrading and that it is there are smaller and smaller fragments chipping off from it. So that is why you have a big bang and then smaller fragments running all over the gel. So a smear on the gel is indication that you have fragmented genome material. Next, messenger RNA or mRNA population can be isolated from the RNA pool using blank affinity chromatography. The options are poly A, poly C, poly T, or poly U. You can put what you think is the correct option in the chat. Devadrita says that we can use a poly A affinity chromatography to separate mRNA. Sejuni says it is a poly U. Okay, so let us look at it. So this is again part of Northern blotting, where you look for your particular RNA samples, particular RNA fragment in a pool of RNA samples. The steps very similar to Southern blotting. The first is mRNA isolation or total RNA isolation. Then next, what you do is separate mRNA fragments on an agarose gel, transfer to your nitrocellulose membrane, and use a cDNA probe library. cDNA is also known as complementary DNA. This cDNA is essentially complementary to your mRNA of interest. So you can make a probe out of the cDNA, and then the probe is radio-labeled again, so you can hybridize and then do a autoradiogram to detect the probe as well as your RNA of interest. So this is what our mRNA looks like. We have gone through this in previous lectures as well. So this is a eukaryotic mRNA. From the 5' end, we have a 5' guanine cap, which prevents exonuclease activity, a 5' untranslated region where translation has not started. Then you have the coding sequence or the open reading frame where the starting codon is AUG that signals the ribosome to start adding your uh, amino acids to make a peptide using the information from the coding sequence. Then you go through the coding sequence and you end up at the stop codon where the ribosome disassembles and your protein is released. A 3' untranslated region that the ribosome never sees. And finally, a poly A tail. A poly A tail is this huge stretch of adenine bases which are added at the end to prevent exonuclease activity again. So the exonuclease activity from this side will have to reach 200 or more adenines to actually reach actually important information in the mRNA. So by that time, the mRNA would be translated. So this is one of the functions of poly A tail. The other can be regulation of how the mRNA is used for translation. And another thing that we use the poly A tail for is to isolate or to isolate mRNA. So when you isolate RNA from a particular cell or tissue, you obviously do the process is known as trizol extraction. We have done that as well. So when you do trizol extraction, you actually get all the RNA in the cell. This will include the tRNA, the ribosomal RNA, as well as the mRNA and smaller other RNAs. The major important fact here is that mRNA forms a small portion of the total RNA present in the cell. The majority one will be the ribosomal RNA and followed by the tRNA. And then your particular mRNA of interest would be, say, only a dozen copies out of that huge pool. So the first thing that you need to do is just get the mRNA from the total RNA and separate the ribosomal and the tRNA out. So you can use the unique properties of mRNA, which we have seen are the 5' cap and the poly A tail. What we use is the poly A tail. If you have a affinity column which has a lot of T's, so a poly DT affinity column, then that will very easily base pair with this huge poly A tail. And since none of the other types of RNA, the ribosomal or the tRNA, have a poly A tail, then you can just separate out mRNAs and have them bind in the affinity column to your poly A tailed column or beads or whatever you're using to isolate, and essentially you have isolated the mRNA from everything else. Then you can wash it multiple times and then isolate just the mRNA from it. So the nucleotide A forms two hydrogen bonding with T, and this pairing is very specific and only occurs in mRNA because they only have the huge poly A tail. Exploiting this feature, mRNA population can be isolated from an RNA pool using a poly T affinity column, and this is known as affinity purification of mRNA. We'll also go through how northern blotting works. The first thing you start with is cells or tissues. You make a single cell suspension and you end up with cells again. And then you do something known as homogenization from the tissue to, and then different enzymatic processes to form a single cell solution. And then pull or put all of this into something known as an affinity column. Affinity columns looks very similar to a Eppendorf if you have ever seen it, or a MCT tube, and it will have a column which is made out of different materials such as nitrocellulose or dextran, which will have these oligo DT beads. These oligo DT beads are essentially single-stranded DTs, so deoxy, thymine, and a long chain of them, which are just waiting for mRNA to pass through to hybridize to the poly A tail, and then you wash multiple times and then you can elute it. So release of total RNA either by lysis buffer using a detergent or homogenization in the case of hard tissue. Mixing of poly T containing beads with the total RNA preparation. Due to mutual exclusive affinity, mRNA binds to poly T beads and every one of the other type of RNA will just fall through. Wash the beads with washing buffer to remove non-specific cross-contaminating RNA or anything else that is bound to the column. And then using an elution buffer, you can elute the mRNAs and its purity can be checked on the polyacrylamide gel. So here, once our mRNA is bound to the column, you can wash using the wash buffer. The wash buffer has majorly it is made out of ethanol. Again, for the same idea that uh, if you have anything that is non-specifically binding to the non-specifically binding to the column, which in this case can be salts. So this salts, even when ethanol, a lot of ethanol is passed through, they will still remain dissolved in solution. So essentially, when you pour a lot of wash buffer on top of the tube, and the liquid comes out of the bottom, a lot of the salt will come out with it. So this you do multiple times to make sure that you remove all the salts that may be contaminating of your mRNA. And finally, you add an elution buffer. Elution buffer is made out of something known as chaotropic salts, chaos-inducing salts, such as guanidium nitrates, which actually increase the entropy in the column. So this essentially causes anything bound to the column to just be released. And when the elution buffer is passed through, all of the DNA bound to the column will just go through, and at the bottom you will have your pure mRNA. So the messenger RNA population can be isolated from the RNA pool using poly T affinity chromatography. Any questions? You can you can think about it for a bit and let me know if you have any questions. Okay, there seem to be no questions. So we move on. In northern blotting, to get a very good separation for messenger RNA molecules, you need to run them on denaturing blank gels. So it will be denaturing polyacrylamide gels, denaturing agarose gels, denaturing starch gels, or denaturing gelatins gels. You can put here whatever you think is the correct option in the chat. Okay, everyone is jumping on top of it and answering B, which is agarose. So let's look at it. So we know for northern blotting as well as southern blotting, we use agarose gels which have uniform thickness of pores to allow DNA or RNA to pass through. Now we look at why denaturing gels are required for RNAs. So RNA gels are always run under denaturing conditions. This is because when we are talking about agarose gels, we were talking about electrophoretic mobility and how the charge by mass ratio is what determines how fast a molecule will move through the gel. But there is also another factor comes into play in case of RNAs. If the so you can imagine a long, heavy, linear molecule of RNA will have a difficult time passing through the gel because has to form essentially a tunnel through the entire gel, passing through and looking for correct pore sizes to go through. Whereas a very tightly bound RNA molecule, which essentially forms a big ball, can pass through very quickly because it can flow through the entire gel looking for pores and it can go through them. So, depending on the secondary structures RNA has, it will, or even DNA has, it can move faster or slower through the agarose gel. And this is especially problematic in case of RNA because RNA always loves to form secondary structures other than the linear normal RNA. So if you compare agarose gel where all of the RNA molecules are linear, then the same size of weight should come up at the same level. But if the RNA is allowed to form secondary structures that will either slow it down or speed up how fast it is moving through the gel, then even same sizes, so 100 base pairs say of RNA will appear at different levels throughout the gel because of how fast they can move. So electrophoretic mobility also depends on the overall structure of the molecule passing through it. So always, whenever a molecule can have secondary structures or different three-dimensional structures which will cause it to flow ununiformly through the gel, then we always denature the molecule to come back to a linear structure or a primary structure. This is seen with RNA, which can form stem loop structures, hairpin loop, pseudoknot, bulge, internal loop, or multi-loop structures, or even in the case, and especially in the case of proteins, which always form different kinds of folds and can have complicated structures and always forms quaternary complexes that have multiple subunits. So you always denature those definitely. And for RNA also, before running them on the gel, you want to denature them. As a result, it gives them less, so because of the secondary structures, it gives them less time for the molecule to interact with the agarose gel and consequently less resolution within the different RNA species. So again, if this is another problem. So if you have 100 base pairs of RNA and you're trying to resolve it, but it can form three different secondary structures, one linear, one hairpin loop, and one a stem multi-loop. Then all three of them will appear as three distinct bands on the same gel, even though they are the same RNA, but because of the different secondary structures, they travel different at different speeds throughout the gel, and thus your one RNA molecule, which should be one, appears as three. And now you can figure out if you have contamination or your RNA has just degraded or what has happened. This is also very commonly observed with plasmids. So plasmids, we have know these circular double-stranded independently replicating DNA fragments that are present in bacteria that contain antibiotic resistance and tons of different other things. They also come in three different secondary structures. One, a normal circular structure that you like to see and that always travels at the correct size that it should. Then you have something known as a nigged plasmid, which has one of the strands or both of the strands broken, which travels way slower. And then you have a supercoiled structure where the plasmid, instead of forming a ring-like structure, it loops and loops, forms a ball and very tightly woven ball that travels very fast through the gel. So the same plasmid will give you three different bands, and then you are confused which is your actual size of the plasmid because you do not know. So in that case also denaturation is preferred, but we don't actually do that. We do different things to figure this stuff out. But in case of RNA or protein gels, you definitely have to denature them. So these are different structures, secondary structures of RNA. Even though RNAs are almost always single-stranded linear molecules, they can loop around and form a double strand. They have they can form a single nucleotide or a trinucleotide bulge in which part of it has double stranded, part of it has not double stranded, and it just juts out. It can form a hairpin loop, which is, I guess, the most famous RNA double stranded structure, in which the base of the loop actually can hybridize and form this kind of a structure, but the upper part absolutely cannot. So it looks like a bobby pin or a hairpin. Then you have asymmetric internal loops, symmetric internal loops, two stem coaxial structures, a three stem junction, a four stem junction. This one we have seen already in tRNAs where they have different arms. Well, there are different arms to the tRNA, and they do different functions, and they have a four stem junction loop. And so essentially, you have a lot of different things that the RNA can do. So, but how do we solve it so that we have one kind of we have a uniform way of figuring out what the size of the RNA is? That is, you run the agarose gel that contains formaldehyde. Formaldehyde denatures the RNA, causing it to not form any kind of double-stranded structures and form a linear RNA molecule. And the linear RNA molecule will obviously run at the correct size it needs to, and you can then figure out everything you need to figure out about the RNA. And it also prevents double strand formation of the RNA structure. So once you remove the RNA and it is in the gel, it will never come back together into a secondary structure, and you have solved all your problems. So in agarose gel, to get a very good separation messenger RNA molecules, you need them to run on denaturing agarose gels. Any questions? Okay. Next, the RNA sample and agarose gel contain blank to denature the secondary structures present in the RNA. The options are ethanol, acetic acid, urea, and formaldehyde. You can choose whatever you think is the correct option. Srini says it is D, formaldehyde. Anyone else? Europe also says it is D, formaldehyde. Chanda also, there is formaldehyde. They also say this D formaldehyde. So we have just seen you RNA secondary structures make a lot of problems for running RNA gels and getting uniform bands. So you add formaldehyde to denature the secondary structures and have a linear RNA molecule running to the gel, and then you have everything. Then you can compare everything very easily. So it is formaldehyde. The next question is blank is used for preparation of RNase free water. The options are Dep-C, DMSO, ethanol, and formaldehyde. Okay, so we have a lot of options coming in. Uh, Srijani Chanda, Oropi, and Vivek, all of them said Dep-C is used in the preparation of RNase-free water. So let us look at it. So whenever you are doing a northern blot or working with mRNAs or RNAs, you definitely need to get rid of RNAs from everywhere else. RNAs are enzymes that degrade RNA and they're present absolutely everywhere. Your body prepares by itself a lot of RNAs and it is present essentially in the air, water, everywhere else. And that will cause a lot of stuff to mess up when you are doing experiments with RNA. So after you have isolated mRNA and even before it, you should make sure you are working in an RNase free environment. But once you have isolated your mRNA, where will you store it? Normal molecular biologists and everyone else, as well as me, we do not trust your normal milieu or autoclave water for storing mRNA. We do something known as Dep-C treatment. So Dep-C is diethyl pyrocarbonate, which is a molecule which can bind to different enzymes having histidine or serine or tyrosine groups, and essentially by binding they inactivate the proteins. So if there is any chance of RNAs being present in your water, Dep-C will go in and bind to your enzyme and prevent it from functioning because it binds to it. And Dep-C is very often used to make RNase-free water, and that water is used to make RNase-free buffers or reagents that can be used in all of these experiments. So how does Dep-C work? So Dep-C has this mm, crooked structure, which looks like this. And each one of these groups can actually bind to different side chains of proteins. It especially binds to histidine, and then due to this binding, it causes cross-linking between the protein and essentially mess up the entire protein's function by folding them incorrectly and binding to important enzymatic residues. It can bind to tyrosine, serine, as well as threonine. So it just binds to everything and prevents the proteins to function properly. And another important use of Dep-C is, so one, you did something great. So you added Dep-C to your water, and now all the proteins and all the enzymes present in the water are now inactivated You did it specifically for RNAs, and now your water is RNase free because now all the RNase is inactivated. Very good. But the next moment when you use this particular RNase free depth C water to elute your mRNA, and then do a next reaction. Say you want to do an in vitro transcription reaction. So you add a DNA polymerase or reverse transcriptase to the water, and then the water has DEPC, and that DEPC will act on your reverse transcriptase and inactivate that. So essentially, any kind of biochemical reaction cannot be done with the water that is made with DEPC, because any enzymes you add, it will be completely deactivated. The idea here is that depth C was specifically chosen because it is heat labile. The entire water, what you do it is you put it in a bottle and autoclave it. The moment you autoclave it, any residual depth C that has not bound to any proteins present in the water will actually just break down into carbon dioxide and water and just be removed from the bottle. So essentially, any residual depth C will be removed when you autoclave it. So you can use that water with any other biochemical enzymes after they have been autoclaved, because now you are completely free to use it, and there is no more depth C that will inactivate your enzyme that you add later. Has already inactivated anything the water had, probably RNAs and anything it previously had. So that is how you make the RNase free water, and then you autoclave it so that you don't have residual depth. So depth is used for preparation of RNase free water. Any questions? Okay, next. Bromophenol blue is the blank dye used to monitor the progress of gel electrophoresis. Is it the loading dye, the steering dye, the tracking dye, or the binding dye? You can put in what you think is the correct option in the chat. Region is C. Chanda and Vivek also say it is C. Tracking dye. Anyone else? Or also says it is C. Tracking dye. So let us look at it. So we have already seen what the DNA loading dye is made out of. First, I had said it is made out of a high density reagent such as glycerol, sucrose, or 5-col 400. What that does is that when you load the DNA into the gel, because of the density, DNA will try to float out of the gel and move into the buffer. You want it to stay at the bottom of the well so that it moves through the gel, not over it. So and the stay in the gel so you can see it. So you add a high loading dye which has a high density reagent which essentially catches hold of the DNA, pulls it to the bottom so that it travels through the gel. Next, you add something known as a tracking dye. So as we have been talking about it, ethidium bromide or any of its alternatives such as cyber green are used to visualize the DNA. As we had seen in the previous image, the DNA was glowing purple. But that is only when you directly illuminate it with UV light, and UV light is not is harmful for us. So you do it in closed chambers with protection and stuff. So when the DNA, when the gel is just running, how do you determine if your gel has run enough or run too much or run too little? So you need something that will let you see the gel even in normal light under normal conditions. So that is a tracking dye. The most common one is known as bromophenol blue, which gives the dye the characteristic blue color. So when you run the gel now, bromophenol blue molecules will form a blue colored, uh, it will be blue in color, and they travel much faster than DNA molecules. So if, upon looking at the blue line, and this is known as the dye front, you can determine how much your gel has run. So when the gel has run up to here, the blue line is right about here, you can determine, yes, the gel has run enough. There is my DNA has run all this way, and probably now I will have good resolving power or good resolution of the DNA strand because it has run all the way it should. So you always take a notice of this blue dye front, and you make sure that it never goes off into like over the gel. If it you have no blue dye front in your gel, probably you have run the gel for too long and the dye has gone through into other places. Like if your dye has moved out the gel, probably your DNA has moved out the gel too. So this is a very important factor to notice if your gel has run properly or not. You want your dye to be right here, which will determine that the DNA is probably up here and it has run through the gel as much as you wanted it so that you have good resolution. If it is here, then it is not run enough, it has not run enough. You let it run for some more time, and it is if the dye front is gone, then probably load the gel again. And the other things that the DNA loading dye has is tris chloride to maintain a proper pH in which the DNA will be very safe and not from any not undergo denaturation or anything like that because of the pH being perfect. EDTA is a chelating agent which chelates divalent cations such as magnesium, which are important for DNA's activities or DNA modifying enzyme activities. So EDTA, if it is there, then it removes all the cofactors and now your DNA is safe, even if there is contamination from any other enzymes. And if you want a denaturing DNA gel, then you add STS, which is optional. So bromophenol blue is the tracking dye which is used to monitor the progress of gel electrophoresis. Next, after resolving the gel, a thin layer of organic solvent such as butanol or isopropanol is layered to stop the entry of water, nitrogen, oxygen, or carbon dioxide. Some context for this we are talking about Western blot gels which are made out of polyacrylamide and not agarose. And this is something that we do when we make the Western blot gel, which is made out of two individual gels. And once the lower layer is being formed, we add butanol or isopropanol to the top. And why do we do that? The answer is to prevent interaction with water, nitrogen, oxygen, or carbon dioxide. Srijani says it is C, oxygen. Odopi also says it is C. Chandra also says it is C. Oxygen. Anyone else? Yeah, Vivek also says it is C, oxygen. So let us look at it. So now we are going to talk about Western blotting. This is the third thing we were supposed to talk about today. So how does Western blotting work? Very similar to Northern and Southern blot. You isolate the total protein from your cells, tissues, or whatever, and this time you load it onto a vertical gel. So previously you have seen the gels are horizontal; they are standing flat down. Now you load a vertical gel in case of Western blot, and you will see the reason for it in a bit. And because of the and then you let again you do gel electrophoresis and allow the proteins to travel downwards, and then you probe using antibodies. So antibodies, you know what antibodies are. These are proteins which can specifically bind to a region of a protein of interest. So antibody against protein A will only bind to protein A and nothing else. Now using this you can detect the protein of interest because only that will have the antibody bound to it. Now, there are multiple nuances to Western blotting. First is that instead of having an agarose gel, you use something known as a polyacrylamide gel. Polyacrylamide, the pore size is better for proteins to travel through. The other thing is we have been talking about how electrophoretic mobility depends on the charge by mass ratio. So, okay, so for DNA, all DNA molecules are uniformly charged because they have one phosphate negative charge per base pair. But proteins do not have that. Proteins have various different charges. A protein can be neutral, acidic, basic, negative, positive, depending on the, and that also changes depending on the pH. So how do you do gel electrophoresis on a protein? Because obviously, when you attach the negative to one and the positive to one to the other, positive proteins will travel on one side, negative will travel on the other side. You will never be able to figure out what the weight of the protein is. So what you do here is that you actually use you denature the protein using SDS. SDS is a detergent which is sodium dodecyl sulfate. Yes, sodium dodecyl sulfate. And what it does is that it breaks down the proteins into the primary structure, so a long single strand of protein, and again it binds through the protein. And SDS has a huge negative charge on top of it. So once it binds to the protein, it causes all proteins to be uniformly negatively charged. So once they're uniformly negatively charged, the only thing that matters is the weight of the protein while traveling to the gel. Now, there are situations in which that doesn't happen very well, but essentially that is the idea behind SDS denaturing polyacrylamide gel electrophoresis. Polyacrylamide for proteins, SDS to denature the proteins as well as mask the negative, the charges on the native protein with negative all around so that they travel uniformly and can be separated on the weight of the protein. The final thing that you can keep in mind is that the probing mechanism with antibodies. So antibodies, as I said, these are proteins which will bind to a specific region of a protein of interest. So if you have a protein A, it will bind to this 200 to 300 amino acids of protein A very specifically. But antibodies are very expensive in nature. So when you use an antibody, you cannot radio label it directly. So how? So if you have a protein, if you have an antibody against protein A that in the world only you use, so making of that antibody along with a radio label tag will be immensely expensive for you. What instead you do is that you make an antibody that is specific to your protein that is way cheaper, and you make a secondary antibody, a second antibody that binds to your primary antibody. And this secondary antibody can bind to a wide variety of primary antibodies depending on which host it was raised in. So if my primary antibody was raised in rabbit, that my secondary antibody against rabbit will be able to detect it no matter what. And the secondary antibody now can be commercialized and sold to a lot of people because all of them, a billion people have rabbit antibodies, which are detecting different things. So essentially, use a secondary antibody that actually has the radiolabeled probe or the fluorescent signal so that you can detect the primary antibody. So probing for Western blot occurs in two different steps: a primary antibody binding to your protein of interest, a secondary antibody binding to the primary antibody and actually giving you signal. So that was a lot of information. I will just quickly go through the basics of Western blot. So Western blot is a technique used to detect specific protein present in the crude lysate or homogenate. It uses the separation of different proteins on a gel electrophoresis using SDS page. So SDS polyacrylamide gel electrophoresis, and then the transfer of protein onto a solid support such as a nitrocellulose membrane. Primary antibody to detect the protein of interest and the secondary antibody to detect the primary antibody gives a colored or a chemiluminescent signal. So now we come to how to make the gel, which is another thing. Does by now anyone have any questions regarding the how Western blot is done? Then we get into details of how the gel works in a Western blots. Okay, so we look at the gel. So the gel is made out of, as we have seen, acrylamide, which is a monomeric unit of the used to prepare the gel, and two other interesting molecules. One is TEMED, and the other is APS or ammonium persulfate. So acrylamide, as you have seen, agarose, when you hear, normally does not dissolve in water. You heat it up and then cool it down, and then it forms crosslinks. Acrylamide actually does not form crosslinks by itself. The cross-linking reaction needs to be started off by oxygen radicals, and these oxygen radicals are produced by a reaction of TEMED and APS. So essentially, if you want to make an acrylamide gel, you mix acrylamide, you mix APS, and then you can put it aside for a bit, and when you are absolutely ready to make the gel, you add TEMET to the mix. And what TEMET does is that immediately starts solidifying the entire structure, because acrylamide will now start cross-linking and forming the gel-like structure. So these three together are used to make the gel. And then you have bisacrylamide, which is isomer of acrylamide, and this forms cross links for polymerization of acrylamide monomer to form gel. So acrylamide can form very easily very long polymers, so stretches of long strings. Bisacrylamide actually forms the other ends. So acrylamide forms long horizontal strings. Bisacrylamide joins these strings. Without bisacrylamide, you will not have a gel-like structure. It will actually Bisacrylamide actually causes the entire thing to come together as a gel. And depending on the concentration of the base acrylamide, you can make even tighter gels which have even limited pore sizes, which will cause even higher resolution of the protein that you are trying to pass through the gel. So how does the acrylamide polymerization work? So this is how the entire polymerization matrix looks like. These are the horizontal chains I was talking to you about, these ones, and this is the bis acrylamide forming the crosslinks. So ammonium persulfate in the presence of TEMET forms oxygen free radicals that induces the polymerization of acrylamide monomer to form a linear polymer. These linear monomers are interconnected by cross-linking with bis-acrylamide monomer to form 3D meshes with pores. The size of the pore is controlled by the concentration of the acrylamide and the bis-acrylamide in the gel. In a vertical gel electrophoresis system, I want to first show you this. No, nothing. I will go to that. In a vertical gel electrophoresis system, we cast two types of gels. So when you are trying to run Western blots, you actually cast two gels, not one. They are the stacking gel and the resolving gel. We'll go into why for DNA you need one gel, for protein you need two in a bit. First, the resolving gel solution is prepared and poured into a gel casket, which is vertical in nature, so you just pour it in. A thin layer of organic solvent such as butanol or isopropanol is added, so to prevent entry of oxygen. So oxygen neutralizes the free radicals and prevents polymerization and causes the top layer to be crumbly. And when you add the organic solvent, the top layer becomes smooth as you need. After the dissolving gel is polymerized, you add a second layer of stacking gel on top of it, and on top of the stacking gel, you actually add the wells where the DNA will be, proteins will be added. And so this is what it looks like. So it is a vertical system. So you have the gel cassette, you put it into the electrodes, and then you fill the entire thing with buffer. You add your electricity, and then the gels will move up, down. And then you have separation of the protein bands. So why do we need two different gels in Western blot? So when you load the sample, which is your protein, in a vertical system, they always so it always depends on how well all of the proteins are accumulated at the end of the gel. So if imagine you're adding your protein here. So different proteins will have different tendency to actually fall to the bottom and appear right here. So you can imagine it as being on a racetrack. On a racetrack, if you have, so essentially gelatin process is like running a race of different proteins. So for straight races, you always see that the runners line up on a single line so that they all have a like the same starting position. So you can figure out how well they are moving through the racetrack. The similar thing appears in Western blot. You do not want a big, huge protein to fall to the bottom of the gel first, and then when you start the gel, that flows very well. Whereas your small protein is still floating at the top and has to travel all through the first part of the gel as well as the second part of the gel. So you make something known as a stacking gel. So as the samples are filled vertically, there's a distance drift between the molecules at the top and the bottom. This problem is taken care of once the sample is run through a stacking gel. So what is a stacking gel? So you have two separate gels. So one at the top, one at the bottom. The idea with the top is that it will cause everyone to come to the same starting position before you start resolving the entire gel. The stacking gel has a different pH. It has a different pH of 6.8, and the solution that you use to load the proteins has glycine, and obviously the buffer has tris hydrochloride. So when in the stacking gel, when you turn on electrophoresis, the glycine is actually moving slower than everything else, and tris hydrochloride is moving at the bottom. So essentially, your protein is sandwiched in between and flows at the same speed to the starting line, which is at the end of the stacking gel. At the end of the stacking gel, the resolving gel is there. So they are two gels, one on top of the other. And at the resolving gel, the pH is 8.8. So glycine becomes charged and it now moves faster. So glycine at 6.8 is neutral. At 8.8, it is charged. So now it will also move and just shoot through the entire gel. And your protein is now left to move under its own speed. It moves fast and now the sample runs as their molecular weight. Due to the SDS, they have equal negative charges. After tracking dye reaches to the bottom of the gel can be taken out with from the glass plate using a spatula and stained with comassive brilliant blue R250 dye, which stains all the proteins, and you then should see bands like this. The dye stains all the proteins on the gel. So this is why you need a stacking and a resolving gel. So after dissolving the gel, a thin layer of organic solvent such as butanol or isopropanol is layered to stop the entry of oxygen. Next question. Ammonium persulfate in the presence of blank forms oxygen-free radicals and induces polymerization of acrylamide monomer to form a linear polymer. So in the presence of what does ammonium persulfate initiate the polymerization reaction? Bisacrylamide, formaldehyde, butanol, or TEMED? Chanda says it is D, TEMED. Anyone else? Auropi and Vivek also say it is timid. Anyone else? Srijani also says it is timid, which initiates the reaction for polymerization. Okay, so let us look at it. So we have already seen ammonium persulfate with timid forms the oxygen-free radicals required to initiate the acrylamide polymerization reaction. So the answer is timid. And finally, not the last question for the day. Northern blotting is used to analyze the blank activity of the gene. The translational, the replicative, the transcriptional, or a post-transcriptional activity of the gene. What is Northern blotting used for? You can put in what you think is the correct option. Chandu says transcriptional. Anyone else? Okay, Srijani, Chanda both say it is C, transcriptional. So we know that when we are doing a lot of lot, we are looking at mRNA and how much mRNA you have in your sample and which mRNAs you have in your sample. So mRNA is obviously going to be will show you how what the transcriptional signature of a cell is. So if so, this is an so all the cells that we have in our body actually has the same exact genomic DNA. But why does our eye look so radically different from our heart? That is because even though we have the same genomic DNA in all our cells, the mRNA that are expressed from the genomic DNA are widely different between different cell types, which are myocytes in our heart or retinocytes in our eye. So, depending on what mRNA are getting produced, the cells do various different functions, and obviously, different mRNAs are expressed in different situations for to meet the different functional requirements of the cell. So, to find out if your particular mRNA is being expressed, which you do with a northern blot, you're essentially figuring out the transcriptional nature of the cell. So the transcriptional output of the cell or the transcriptional activity of the cell as well as the gene that you are investigating is investigated using Norton. Okay, that's all I have for you today. So thank you for joining. And if you do not have any questions, then we can end the class right here and you guys can leave. Okay, thank you.